Okay, it's my pleasure to introduce Anu Kalimo. Anu, thank you very much for coming from Finland. And Anu is a very famous neuropathologist, and in my opinion, he did some very important contribution in the field, not only of disease, but also in the field of other small vessel disease of the brain. So thank you, Anu, and try to keep on time.
Yeah, thank you for invitation, and also thanks to the foundation.
Take the microphone and
Yeah. And which?
Pointer.
Yeah.
So the pointer and to move the slide.
Yes. Yes, thank you to the organizers and also for the foundation. My mother was a dressmaker, and she also had a Singer sewing machine. Okay, let's go to the substance. We have several of these small vessel diseases, and there's a list of several of those, and I will present at least some of those, depending on how fast I can be. And it was now clear for you that the problem in CADASIL is the extracellular domain, which does not where you have the mutations, and how does that mutation then affect the tissue, the brain at the tissue level? That is, I'm a neuropathologist, so my talk is mainly about the morphology of this disease. So how can you take those off, those lights, so that, yeah. How the blood vessels look in the brain of CADASIL patients. You can see there that in normal patient, arteries are quite thin-walled, and in CADASIL they are markedly thicker, especially in the white matter. And it was shown that there is accumulation of this extracellular domain in those arteries, especially in the white matter, but also in the gray matter, and also the target of this mutation is the smooth muscle cells in the vessel wall, which are sensitive to this mutation, where the NOTCH3 is expressed in the adult people, and they are degenerating, and loss of those. You can keep them down all the time. And what happens there? We showed that there is definite decrease in the blood flow, especially in the white matter, already after the age of 30 years. And that means that this blood vessel changes must have definite effect on the blood flow. Even though there was some discrepancies, some researchers, two teams, Okeda's team and Rousseau's team, they showed that there would not be any stenosis, so that the blood vessels were not narrower. But what was wrong in that respect, that they did examine vessels of wrong size, too large, and since this affects the small arteries, and the stenosis becomes apparent not until you come to the size of these vessels of 50 micrometers. Then you see that in CADASIL patients, those very thin or narrow vessels their number increases considerably. And that became, for a pathologist who was looking at these blood vessels, it was obvious that there must be some kind of a stenosis, like that analysis showed. And why this stenosis then leads to infarctions, it was already pointed out here that these vessels are terminal vessels, longer vessels that go to the white matter, and they are terminal in that respect that they don't get any help from their neighbors. The collateral flow does not help there in those at the small vessel level. And the result is the lacunar infarctions. And also something that we are quite convinced, and something that we are now trying to analyze, there is some metabolic difference between the blood vessels, the small arterioles in the gray matter versus the white matter. What the metabolic difference is, that we are trying to find out. So how these brains in patients look when they die, when they have suffered from the disease quite long. And you see that there are many lacunar infarctions, especially in the white matter and in the deep gray matter, the basal ganglia and thalamus, whereas the cortex is relatively well spared, even though we did see that there are some small infarctions, but not any major, or few major infarctions, so that the cortex looks fairly intact when you look at that with the naked eye. And on the other hand, it cannot, CADASIL doesn't protect from these risk factors. Here is a view of a CADASIL patient who has these classical infarctions, but since this patient had also atherosclerosis affecting the small arteries, this patient had a cortical infarction, which was clear. So this extracellular domain accumulates especially in the white matter arteries, which are thickened. This is antibody. Thanks, Anne, for this antibody. And you see that also the blood vessels in the cortex, they have this accumulation of the extracellular domain, but they are not similarly thickened. And also the capillaries, in their walls, you can see the accumulation of extracellular domain. And like it was discussed, that the skin biopsy turned out to be a very handy, a very reliable diagnostic method, and it could be used, the antibody could be stained with the antibody, and you get that result. But since I'm an electron microscopist, sorry, Anne, I rely more on electron microscopy, and we did find that quite reliable, like in the previous presentation you have heard. And it is the small arterioles in the skin, and we select the blood vessel on the base of the toluidine blue sections, so that small arterioles with a couple of smooth muscle cells in the wall, and you can see also those blue dots, which are the fragmented elastin. Those are usually quite representative. So you can have a picture like this. There's a lumen, endothelial cells. There's often a space between endothelium and smooth muscle cells, and you can see accumulation of granular osmiophilic material. Of course, you have to recognize the substance. It does have quite a specific appearance, so that here is a higher magnification of this material. It is usually in indentations and darker towards the cell and really clearly granular. But there are some why I consider electron microscopy more reliable is that at the early stages of the disease, when you have only little of this GROM, it may be that with the immunohistochemist you are not able to detect such a small amount of that. And here, for example, this is the youngest one that I have had for examination, 19-year-old male CADASIL patient, son of a homozygous CADASIL patient that we found in Finland. And when you have very little of this GOM, it is understandable that perhaps the immunostaining is not always positive. So like I said, as an electron microscopist, I think that it's not so complicated. Very extensive sequencing will take certainly as much time as a good EM lab will find and look at this specimen. So I recommend also these skin biopsies like you are now doing also here. But you have those confusing findings, so that you have to know how GOM looks. There is that true GOM, but there are, for example, the elastin fragments may look a little bit similar. There is that, but I don't know what it is. It is granular, it's osmiophilic, but exactly what it is, I don't know. And it may even be in indentations like real GOM, and then also different cell debris that may confuse, so that you have to know what are you looking for. And even this, it's not an absolute truth, but you, at least you get forward. And here are these questions of non-cysteine mutations. I think I have examined myself about 6-7 cases, and none of them have had GOM. But there are reports about this, and some of those pictures that I have seen in the publications, I don't really, I'm not convinced that only this, which is from Mitsuno, that looks at least very close to real GOM. But for example, here, these dark spots, and here, it's a little bit questionable that you should know exactly and analyze. Then, if you have, if it's questionable, analyze several sections. And since we are interested in non-cysteine mutations, we have, in collaboration with a group from Athens, started this analyzing the molecular structure of these. And in notch 3, you have these alpha helices. And if you have a mutation where it is arginine tryptophan mutation, that does affect the beta cell, beta sheet structure, so that it may be that these do have such an effect after all on notch 3 that it does cause the disease, but it is still open. We will continue with that. One thing that was reported by two teams, Lesnik Oberstein in Holland and Martin Dichkan's group in Germany, that they do have often these microbleeds, and we can see them, of course, morphologically. This is one of these microbleeds. Majority of those are in the more or less in the gray matter, in the vicinity, in the deep gray matter, or in the cortex, and you can see these as an extravasation of iron, because the blood cells that are extravasated, they are disintegrated, and the iron accumulates in the macrophages. And with this staining, one can show that there is a leakage. Why they are mainly in the gray matter area, not in the deep white matter. It became, to me at least, I think this is suggestive of the reason that thickness of the arterial wall in the white matter of CADASIL patients, this is elderly and there is a young, it is remarkably thicker if you compare to the controls where they are almost the same size. Here the thickness is about double compared to the gray matter, and they are at the same time most probably they are more prone to leak because they are weaker. The smooth muscle cells are lost, and obviously the vessel wall, arterial wall gets weaker, and there you can see how thin this wall is in the microbleed area that I just showed you. And also that begins already at the young age. There is the youngest that we have had, 32-year-old patient, already there. The thickness of arteries in the white matter is increasing. And something that we were analyzing, we also at the same time found an interesting that we had not thought before, that the luminal diameter of the white matter, if you look at the controls, elderly controls, they are markedly larger in the white matter than in the gray matter, so that the blood has a wider route to go to the white matter. But on the other hand, in elderly CADASIL patients, it really shrinks. It becomes smaller than the diameters in the gray matter, and with this coefficient, one can see when it approaches 1, like here in the young patient already, it's approaching 1, and when it goes below, then that speaks for pathological arteries. And it was discussion, do these leak, these blood vessels? We have used fibrinogen as a marker for vasogenic edema. That means the blood proteins leak from the blood vessels. And those thick-walled white matter arteries, they don't seem to be leaking, at least very much, because here is a fibrinogen staining from a hypertensive patient, and you can see how they leak to the surrounding tissue. Of course, when you have a microbleed, then you have also fibrinogen surrounding the vessel, but these don't seem to be leaking so obviously. But then, on the other hand, the chronic effect of this vasogenic edema can be seen when you stain the cerebellum, because the proteins that extravasate cerebrospinal fluid takes that around cerebellum, and the Purkinje cells take up this fibrinogen, and like there you see a patient who had epilepsy, that the Purkinje cells are strongly positive because they have taken up fibrinogen from CSF. And in the control, on the other hand, they are negative. And here are examples of two CADASIL patients. One of them had quite a few of those positive cells. Another one had only weakly stained, but it looks that there is some leakage, some kind of a chronic leakage, perhaps from the gray matter areas where you have these microbleeds, for example, and so on, that there is some damage to the blood-brain barrier. And okay, so this was the main findings in CADASIL. Then if I will go to the next one, when the first workshop was here in Paris, Patrick Surander, who was the who had discovered the Surander-Volinder disease that was published under the name Hereditary Multi-Infarct Dementia in 1977. He was here, and it was thought that this was the first CADASIL published. But then I got some of the samples from this friend of mine. He's my teacher, he's my senior teacher, and I was surprised not to find, for example, the typical changes in the skin biopsies or electron microscopy of the brain tissue, so that there was no GOM, even though the findings were quite similar to what you see in CADASIL. Lacunar infarctions in the white matter, also small lacunar infarctions in the brainstem, so it was understandable that that was suspected to be CADASIL. But then when this was continued, together with the Ras Kalarias group from Newcastle, we have analyzed these further, and the difference between CADASIL and this Swedish MID, it was published in Brain 2007. There are certain differences, but they are not so striking, the clinical picture. But then the one thing that is quite important, that there is no GOM. Here is a CADASIL electron microscopy, and there is from one of these Swedish patients, and extensive search for GOM was unsuccessful, so it was no GOM. And also the staining for this extracellular domain, that was negative in this Swedish case. And also the clinical picture is to some extent different. And in Newcastle, they also analyzed 4 members from the Swedish family, the NOTCH3 gene, and did not find any mutation. So this is another disease. And its gene defect has not been found yet, and we are still in the process. We have collaborated with those from Gothenburg, and there is a genome-wide analysis in process, and on the basis of the friendship of one of our members in our team, John Hardy's group is now trying to find what the gene defect in this disease is. Also, that at the blood vessel level, there are some differences, but on the other hand, on the basis of those alone, it may be difficult to identify, so that electron microscopy would be perhaps the best one to differentiate between these two. So that was that Swedish hereditary multi-infarct dementia. So you will hear in the near future, I think, what the new gene there is. Then a few words of the very common arteriolosclerosis. Majority of those cases are sporadic, and that affects also the mainly the white matter blood vessels. And here is, they are, many of those patients are hypertensive, so that the small arterioles are affected by high pressure, and that is another additional burden on those. And one could see that in some of those at early stage of their disease, you could see that the smooth muscle cells in the, like here, it is strongly stained. It tries to kind of enforce its walls, whereas in this larger one, the smooth muscle cells have started to degenerate already, and accumulation of collagen 1, that means fibrosis of these small arteries, has started. And that will cause later on quite marked stenosis with only a small lumen remaining, or almost a total occlusion. On the way from these changes to these end stages, one can see that this hypertension obviously makes blood proteins to leak into the vessel walls, and then you see something that is called fibrinoid necrosis of those walls, and it is followed by this additional deposition of collagen and fibrosis. And how they look, the lesions are fairly similar as seen in CADASIL. That means that they are mainly in the white matter, and they are lacunar infarctions. When it's quite extensive, you can see on the surface already that there's this kind of granular cortical atrophy. The surface is not smooth, and in the microscopic sections you see small infarctions which are fairly small in size, up to of the lacunar size. I don't say many words about the atherosclerosis, because the common atherosclerosis, which you all, I'm sure you know that disease, that can also creep into the small arteries within the brain. Like here, you can see that this was the case when I showed in this CADASIL patient that there were small arteries which had marked atherosclerosis, and then it causes formation of plaques in these walls, and then stenosis, and end stage could be the occlusion. But I don't go to any further details. A few words of this cerebral amyloid angiopathy, which is also included among the small vessel diseases, and usually the most common molecule that accumulates in the small artery walls is beta amyloid. Very common in Alzheimer's disease patients. Almost, I think, 80% have at the same time this amyloid angiopathy. And there are also, besides sporadic cases, several hereditary forms of this disease. And how they look, they are similarly rounded with somewhat thickened walls, not as much as in CADASIL, but there it's stained with a Congo red, it's bright red, and it has a birefringence with the polarized light, and it is positive with the fluorescence of thioflavin, and they may even have this kind of what is called the double barreling, that the inner and outer parts dislocate from each other. And that amyloid angiopathy, it is different from these other diseases now that it is mainly the disease of cerebral cortex. So that in the white matter you see very little actually. So when you look at this, this is from a hereditary case of Arctic mutation, Alzheimer's disease. You can see how the penetrating artery is positive for amyloid, but when it enters the white matter, it stops. So it is a gray matter disease. And when you look at electron microscopically, those what you have accumulated, what the disease accumulates in the blood vessel walls, there are plenty of these microfilaments of amyloid. And what does this cause? The blood vessels, they don't become stenosed as do in Cadacil. Instead, these are now, the outer diameter is the same, but here the blood vessel wall is much thinner, and since there the smooth muscle cells have degenerated, it's mainly amyloid and some extent collagen, and these seem to be prone to rupture, and that is typical for amyloid angiopathy, that they get these hemorrhages, brain hemorrhages, and they are typically of the lobar type. That means that from the area where this is prevalent, this disease, that means from the cortical area they break, and the bleeding is lobar. That means it's not in the center, usually not in the basal ganglia like the hypertensive one, but it is peripherally in the brain. And then there is also this accumulates this beta amyloid in the small capillaries, like you see there, and Tal et al. Have analyzed what this capillary CAA does, and it seems to be that it does affect the blood flow. Even though they do not see obvious infarctions, but it does cause flow problems, and that is why it obviously affects also the function. And that shows that, okay, I will jump over and go to the next one. Cadacil will be presented in detail later on, so I will not go to any clinical aspects, but show a few pictures from these cases. Also there, the blood vessel walls are, they suffer from this disease, so that they become thickened, and also they may have this double barreling, and that causes the problems with the cerebral blood flow in their brains. And that was reported from Asia previously, but also in Europe now, one family at least has been found. And then Padmal, which is something that may be close to this Swedish type disease, and that means that in their blood vessels you see similar thickening of the walls. There is accumulation of collagen and loss of smooth muscle cells, and that leads to lacunar infarctions, like you see here. And then finally, just a few pictures from this retinal vasculopathy. Again, you see small infarctions in the brain, which are due to this TREX1 gene defect. And here, then, I can summarize that the problem with these diseases are small vessel diseases. It is the cerebral blood flow, because otherwise, what is the mechanism behind why the blood vessel wall is affected? that is the problem that you have to solve for each of these diseases. But the common factor is that the flow decreases. And here's our team, and I have usually I end my presentation by showing this. I am from Turku, where we found first the Finnish family, and where our team was started and is still to some extent located. And why Turku has been such an important in this field is clear from this slide. Thank you.